this mass spectrometry bioinformatics proteomics meeting, and actually I couldn't remember, but I said, well, we spent a week, and then I looked at his picture and said, of course, we spent a week there. So, but he was very fast and effective in organizing this, because I just decided to come over. It's good, yeah, it was fantastic, and also the set of meetings that I had today was very enjoyable. I mean, yeah, yeah. Please, yeah, I'll be.
Nice. Hey, welcome. It's good to see everybody. It's cold out. I hear it's going to snow a bunch on Friday, so we're in the middle of it. It is winter around here. Welcome to winter. Michal Linial is here from. She's the director of the Israel Institute for Advanced Studies. We want to hear about that at some point, and she's a professor in the Department of Biological Chemistry. And they have a life science institute there as well as we do, and it's all in Jerusalem. And Michal is a computational biologist. She directed the Computational Biology Center there for years, the institute. And she's involved in proteomics. She's involved in what I see is kind of, I think what we're going to hear about is a little bit about kind of molecular epidemiology, I mean, frankly. And, and bioinformatics can shed light on that, especially when you're looking at phylogeny and relationships thereof, and, how things like viruses and toxins co-evolve and things like that. So I think we're in for a big treat. And the talk title is Modulation by Hidden Peptides, a Playground for Evolution. And we're so delighted to have you and looking forward to your talk. Welcome.
Thank you so much. Okay. So good afternoon to everybody. It's really a great pleasure to be here. I had a fantastic meetings this morning, and I'm waiting for more for tomorrow. And today, I would like to share with you some of what you see, the hidden part of peptides that we don't talk that much about, but the real topic of the talk is basically on innovation, on evolution innovation. And I'll share with you some ideas regarding the universe of proteins. That will be the first part. And then we'll move to some more exploratory tour on toxin, peptides, immune, and brain function. So it will be kind of a pretty broad tour. So let's get going. So when we talk about proteins, I'm just showing you something that you are all familiar with, how data is accumulating and the speed of doing it. And what you see here is just a graph from UniProt, which is the collection database from protein. And this is 2012. And what you see here is that, first of all, it's picking up very fast. But more embarrassing is this comment that I add, which is like a third out of the 20 million proteins that are accumulated here, a third of it are hypothetical or putative or predicted, which means that we have a huge gap between data, as data, sequences, and knowledge. And I'll try to show you how can we move and improve this a little bit. So let's go 6 years later. Now we are at 2018, and actually we are data from last week. What you see is the same resource, UniProt, now have more than 100 million sequences. And everything that I showed you in the previous slide is this part. You see that it keeps picking up. Even that, at 2015, they decided just to overcome this redundancy to remove 50 million. Let's throw it out. Still, you see that when you go and continue watching, it keeps picking up. So as I said here, there is an endless race for removing this redundancy. But more importantly, if you ask why is it keeping going and keeping up, it's mainly because what we are all familiar with, the genomic revolution and the technology of genomic sequencing, and there are tens of new complex genomes that are picking up. And as you can see, from 2012 and on, this is really large number. And what is the end point of this is that we have much more data. We have somewhat better sampling. We have covering some unexplored niche, but as I'll show you in a minute, it didn't really bridge the gap between data and knowledge. And that's exactly what I want to show you here. What is shown here is this is the 20 million that I discussed before, and this is the 107 million that I discussed now. This is 6 years in apart. And very embarrassingly, if you take this, the 100%, you can see in red is everything that we means the community, ever seen in proteomics, in transcriptomics, in you name it. 90-something%, almost 98%, is predicted, is inferred, we never saw it. Not in mass spectrometry, not in RNA-seq, not in Affymetrix, nothing. So that means that it didn't really change over time. We didn't really match the gap between knowledge and data. However, we have more data, and that should be beneficial to us. How can we do it beneficial? So just to give a little bit of number, the task is really to chart this protein sequence space and to bridge the gap between sequence to function. And if we just stick with this 20 million, doesn't matter, but 20 million that I showed you, and we ignore for this discussion completely the dynamic part of the protein space, which is post-translation modification, alternative splicing. This is just estimate a factor of 5, probably it's a factor of 10, but just for the sake of simplicity, there is a thought that with the homology search, one can get from this number to something around this number, which is homologous superfamily, and maybe even to a protein structural fold, which will have even a 10-fold compression. But this is very hard, this jump. And what I'll tell you today is about this jump. And when you go to protein function, really it's an ill-defined term. And I would say that we know very little how to handle it. Okay, so just to give it as a broad statement. Although, as you can see, there is quite a lot of work that we put over the years on this aspect. So the idea that I'm trying to share with you is that we would like to build a map. And as you can see here, this is an old map, 300 years map. It's of the Middle East. This is Israel, this is Jerusalem. It's not accurate, but no mean. But still, the main features are there. So this is the idea. We want to know that we are going to build a map that is not perfect, but at least it's a guideline for some major feature of this area, of this space. So the idea, the goal is, I already told you how we do it. We want to use only sequence. Why only sequence and not data, not other type of data? Because sequence is what we have for free, in large amount. We want to use all protein equally. We don't care if they are putative, inferred, hypothetical, everything should be in. And we want to use an automatic, unbiased method. And the idea is really that homology derived from a common ancestor, and that's the only principle that we keep. So the implementation of what I just say in high-level words is a program called, or a tool and a database called ProtoNet, which is a bottom-up clustering. And the only principle that is in this scheme is the following. First, we start in which each protein sequence, never mind we know it or we don't know anything about, is checked for its distance, let's say, by BLAST e-value to any other protein, which means billions of pairs. So it's a huge matrix. But once you have this, we use some bottom-up agglomerative algorithm, and we refine the merging rule to say when two clusters needs to be merged. And then we prune the tree to leave only informative cluster to remain. And then we do validation and quality control. So it's a very, high-level story. But we should remember one thing, and I want to make one moment on this item. We know that among this 107 million data point that we have today, about 0.5% are really of a good quality. Very good annotated, but they are tiny, right? So the question is, how can we use this tiny, good, manually annotated to improve this? You see, so that's the challenge. So I'll give it in a very naive way, but very intuitively. Let's say this is our protonet clustering scheme. And this is the interaction, let's say the blast value of proteins within the Swiss spot. This is the small portion. And this is another one. But you can see that there are some edges here that are of low quality. Are they real? Are they noise? Right? This is the problem. So what we want to do is we want to benefit from weak similarity between remote families. And the answer is that we add now all the 100 million proteins that we know nothing about. However, as you can see in this A and B cluster, there are a lot of weak but consistent similarity among items in this two-sided, right? So the signal is amplified because we see consistent similarity. So by that, in a very broad sense, we improve the signal over the noise. So let me show you in a very, illustrative way. What we did is this. We boosted A and B such that we can have some proof that they are related. So how can we know, okay, we did it, but are those any good? I mean, you emerge A and B. How do we know that it's worth something, right? So the way to do it is the following. We take each and every protonet cluster that we have in this tree that start from millions of items and end up in one root, right? So, and we take an expert system. So the expert system can be one of many. For example, the structural resource of CAT or SCOP or any of those. And we compare these two entities. And of course, we have the true positive, the false positive, and so on. So we can have a measure. We call it corresponding measure, which is basically the Jacquard score, just the how much. So when you have 1, it means that the two fully agree. When you have 0, it means that there is no match between the two. It's a very intuitive. It's just the size of the intersection divided by the size of the union. So we do this, and we check. And the experts outside are many. There are tens of experts. For example, Go annotation, superfamily, G3D, SCOP, CAT, EC enzyme, PFAM, Interpol, you name it. Every expert system can be checked. And now we can have some quality control to what we did automatically, right? That's the idea. And when we do this, we basically want to understand if this is a legitimate cluster or we have to stop here. So basically, what is the granularity of this superfamily? Because as I said, we start with proteins and we do a bottom-up clustering very carefully, but we go to the root and at some point it's become to be garbage, right? So what is this? And to do this, I want to mention another very critical point, which is the trap of false transitivity. Again, it's a very intuitive, so I'll do it very fast. If you have a protein, most proteins do have more than one domain. If you have this one, and you have this one, A and B, and you have C. So by looking at BLAST, you have a very good correlation between the A and B, and an extremely good between B and C. But A and C are, no matter, they are not homologous. You can see this. This is just a chaining, of the domain. So A and C are not not supposed to be homolog. So they are false transitivity, and actually they do not obey the triangular matrix because the distance between A and C is not smaller than the sum of the two. So we have a problem, right? Houston, we have a problem. So what can we do? So I'll show you in this exactly this example. So this is the example that I showed you before. So let's say we did this clustering in ProtoNet. We did this, and now we have to decide, should we combine this or should we combine this? If we just go for single linkage, you know, simple algorithm, it will probably say this because the value here is much more, it's better. But if we do something on the average linkage, we have many very weak connections, but many of them consistent. We already discussed it. Here, when you do the averaging, the average here is far better than this. So we decided to go on this direction. And we do this. What I'll show you here is a quality control against PFAM. PFAM is one of the top domain-based data source. And everything above this line, this triangle, this line, means that we have higher sensitivity in ProtoNet. Everything below this means that we have a higher sensitivity in single linkage algorithm. So I don't have to show you which algorithm perform better towards PFAM. This is the expert, right? And the two colors means big cluster and small cluster. So we are good for both. So it's not just we are good for big cluster or small cluster, but in general. So this is just the score. On average, that's what ProtoNet do, and that's what the single linkage do. And just to show you that now, let's be more courageous and compare the performance versus 14 expert systems that are combined in Interpol. Okay? All 14. 14 means PFAM, CAF, SCOPE, and so on. When we do this, and so this is the score that I described before, and you can look at some of it. And let's see what is the performance on other known clustering, including, by the way, ourselves with the previous algorithm. So we see that we are outperforming very well this, including our previous algorithm that was a little bit less clever on this. So by this, I hope I can show you that we were able to do quite well with 2 million proteins. So let me give you one intuition. Why was it important? A little bit, I like the analogy saying that, let's say, PFAM giving excellent model for each family or each domain. What we do is we connect them. So each cluster, we have some distance between the others. So you can navigate better on this one rather than on isolated item. This is just to get the intuition. And remember how I started the talk by talking about hypothetical. So this is the size of the cluster of ProtoNet. And you can see that we have able to include a lot of the hypothetical without reducing the quality. So that means that this system really is a very good system to deal with a lot of the hypothetical. That was the point that I want to mention. So what are we doing with this map? I told you that you can navigate, but what, where, why? So one thing that we do is that we were able to participate in the CAFA automatic functional annotation competition, and there are some report here. We were able to find hidden connections that were not known before. We were able to detect faulty annotation because they seems to be uneasy going with the annotation. So this is another trick. And we were able also to understand much better on genome evolution. And this is also, you can read about some genome on this. So to summarize this part of the talk, I showed you that data leads to knowledge, can lead or should lead to knowledge. I didn't mention it, but what we are using is really embarrassingly low plus e-value, 100 or even 1,000. We know that it's noisy, but we don't care. The data overcome the noise. This is a very important point, by the way. When we didn't use it, we were with the less quality, as I showed you before. The navigation of the map is without a map is impossible. You do not like to navigate without Waze. I do not like to navigate in Ann Arbor without Waze. Hierarchical clustering is a strong discovery platform, and update with million and million of sequences remain very challenging. I shouldn't underestimate this. And ProtoNet classify currently 20 million sequence to this number of cluster with very high quality. So this is just to say that we were able to deal with this number. But up to now, I told you about proteins that having sequence or infer sequence function. I already mentioned that there are a lot of maybe and a lot of wrong one. But now I want to take you into a different niche, which is when we have no similarity, no homology, no evidence. What can we do? Let's work it out. So who might be those that fits this, that we have no sequence similarity to use? Those are short proteins. And why do we have a problem? Because from genetic point of view, for example, next generation sequence, when we have very few reads, there is no assembly. No assembly, the data is not in the database. It's not in the database, we cannot say anything about, you see? So it's a little bit of a circle. In terms of proteomics, it's even worse because the best we can find in proteomics in mass spectrometry is when we have low coverage, forget it. And mis-spectra modification kills it all. And the database search, which is the power, let's say, engine of proteomics, is useless because if it do not exist in the database, it cannot be found. So it's a catch-22. So short proteins are really cannot comply with what we used to do. So why do we care about them at all? We care about them because they are easy to create also from evolution. You know, short one. They are easy to produce from biotechnology. It means production. Many of them are tissue specific, like cytokine and toxin. They are novel. Many of them are novel, i.e. they can be found in human but not in rodent. And my hypothesis, which I don't evolve today, is that short proteins are really unexplored but rather common regulatory mode. And we discussed it in the morning with Peter. Yeah. So this part tell us about how we search them, how we develop feature-based predictor, how we go to unseen genome, and find new functions. Okay? So let's do very fast move around this. So we start by saying that we want to use the short proteins, and we start looking at Swiss prot for the same reason that I mentioned before. They are heavily annotated, and we know quite a lot about. So something that I think is very interesting to see, if we bin the proteins according to their size, and you see in orange the fraction of uncharacterized, you see that the shorter the protein, we know less about. So it's an inverse relation between the size and the knowledge, okay? So those are really unexplored niche. So we wanted to know what they do. So we go back to Swiss prot and ask what they do. And you can see that many of the short protein are ribosomal protein, photosystem 2 protein, and so on. But the most interesting are those. Many of them, this is the p-value, extremely enriched in toxin, neurotoxin, and ion channel inhibitors. So for example, out of 2,000 proteins that are marked as annotated as ion channel inhibitor, 1,700 are short. So most of them, almost all of them, short by the size of 100. If I move to 110, it will be all of them. So they are really short. Okay. But very interestingly, they are spread throughout the metazoa world in a very diverse and sporadic way. You know, there are some in Cnidaria, in Bilateria, in arthropod, molluscus, and so on, even in mammals, right? So why searching them? This toxin-like sequences. Again, it's all listed here, but in the recent years, there are a very interesting industry around peptide therapy. And I'm listing here the source of the toxins, the name of the toxin, and what they are applied in the clinics. Okay? So there are painkillers, they are work in immunosuppressant, in chronic pain, and so on. So they are used already as a clinical drug in a field, evolving field of peptide therapy. But what's common among them? So what you see is a channel receptor, and this is the animal toxin, as we call them ICI, ion channel inhibitors. So what's common? First of all, they are all extracellular. They are all short. They are working on receptor, as I showed you. They are stable, extremely stable. Why? Because they have to sit in the snake for half a year till it bites. So they are not falling apart. This is very important. They are specific with high affinity, and they are super potent. You know, this snake that bites an elephant can kill it, right? So here I want to show you some surprise, unexpected. We know, I already told you that if we had a fold of a protein, we know quite a lot about its function. But here, look at this. We have many types of fold, all of them targeted to the same target. So fold by itself would not help you. We have the other case in which the same fold, this is scorpion, have many targets for one fold. So this channel, this channel, this channel, they are working with the same fold. So working in a simplistic way, fold means function, doesn't work. So maybe sequence does. Not true. Let's move. Here are two folds, similar folds. If you look at the sequence of the two, they are working on two very important calcium channels, but only the yellow parts are important for their function. So nothing. Sequence, again, doesn't help. So again, sequence similarity is not a strong characteristic of those. So what could be? If not, here is another example. So if it's not sequence, if it's not fold, let's think what can be unique in those. So a common feature for many of them, or almost all of them, is their stability that I mentioned before, and the compactness. And the compactness is caused by large number of disulfide bridges, like I show you here, like I show you here, like I show you here. This is just an example coming from this conus. So this gives them a superb stability. Sequence, forget it. Fold, forget it. If this is the case, what can we do? So what can we do? We design a machine learning methodology. And in this machine learning methodology, I think many of you are familiar, so I won't go into the details, just to say that you need to have a positive and a negative set, and you build a predictor that can identify new observation that you haven't seen before and give you the right answer. And we do this by taking the sequence, only the sequence. I'm keeping this as a rule. The sequence is the only thing that I have in mind. And we want to learn from the sequence to build up a set of features. But what type of feature? The most important part of this work is to be able to find and represent the sequence by the right features. Which feature? How to capture hidden properties. So let's learn from the expert. And who is the expert? This is a nice snake, very beautiful. The snake toxin, they know what they do. So let's learn what the feature they are using and learn from this. And indeed, this is an example of snake toxins. And we build up over 600 features. Many of them are trivial. Some of them are we engineered. So those are features that try to capture our intuition. So design feature that capture the biological intuition is the most important part in any machine learning. Whoever tells you differently, don't agree. I mean, there are a lot of methods and so on. The methods are less important than the feature. So just build the right feature. So let me give you an example for a feature that we engineered. Let's say this is a toxin, a small one. So what we are doing, we build it in a window of 5, and we ask for each window how many cysteine, or for that matter, how many any other amino acids, and we build this, and we have 32 probabilities. We feed the machine this, okay? So this is the feature that was learned from the sequence, directly from the sequence, but it's not a sequence per se. This is another feature that I kind of like. This, we call it center of mass. We know that in many toxins, like in this one, the N-terminal and C-terminal are very close together. So they're, for the stability, they are not floating around. So if this is the case, let's make as if it's a cycle, and then let's measure what is each amino acid, the distance between this middle way and the center of mass of this cycle. And if you do it to the cysteine, you see that they are spread very nicely, so they are very close to the center of mass. So all those features are fed to the machine, and the machine is working. And let me show you what are the results. So this is the feeding of the machine, and this is the outcome of the machine. What you see that most of the protein are negative. This is the negative. But you have a tail of toxin-like proteins that are positive, and the AUC, the area under the curve, is extremely high by doing a machine learning, tests. So we call those proteins that are here TOLIP, for toxin-like proteins. Okay. So we also design a website in which you can plug in any sequences and ask whether it's a TOLIP or not, toxin-like or not. But the question is, where should we search for them? If you want to search, where? So let me show you how we do it in an unexplored genome of the honeybee. This is a sequence that we find, a novel, unknown, not related to anything. It has a signal peptide, which is a nice sign that it will be secreted. It has a tail that we don't know till now what it does. But when we look at the central part, after we cleaved the signal peptide, we saw that it's extremely nicely aligned with omega conotoxin protein. Omega conotoxin are from the conus that I told you before. So what's the relation between the conus and the bee? There is no connection, right? So when we do multiple sequence alignment and I signed all the shared region, this is the conotoxin, this is the omega conotoxin, and this is our sequence. You see that it fits beautifully, but building this was able to find another set that no one knew about from many different insects and planaria and so on. So this unknown omega conotoxin-like protein, they have quite a lot of homologs that no one knew about. So what this guy is doing, first of all, we figure out that it's expressed not in the venom of the bee, it's in the brain of the bee. In a high, this is a PCR in high level. Second, when we do modeling, structural modeling, all the key elements of the conotoxin are in place. So we ask ourselves, could it be that this works on a calcium channel like the conotoxin? So we express it in bacteria, we fold it, we did everything that is needed. We went to the mass spectrometry and measured the size and so on, and we injected to fish this protein. This is the result. What you can see here is the active OCLP, the oxidized, the denatured one, and some hydra toxin as a control, positive control. And we can see that those fish got paralyzed in a reversible way. After 80 hours, it's a cycle. So this is very interesting because when you inject those guys with the hydra, they die. They cannot recover. So you see one very interesting element. So we took what I just told you, and we took genome, we make them protein, we went to the short, we learn the ClanTox predictor, we go there, we find candidate polyps, and we go to the lab to do functional assay. So this is the cycle for many different genomes, And just to say that in amphibia, there is an expansion of those. In insects, there are enormous collection. We just published this. So for example, in insect, there are almost 400 unknown, overlooked tulip-like. 110 of them, third of them, uncharacterized. No one saw it before. So when we looked at it, let me show you something very interesting. This protein is from the firefly. This toxin is from the human. This is defensin, a very important toxin that works on the immune system, our immune system. This fold is exactly this fold. It just clipped this. It doesn't need this helix. So we can show that this subset is conserved. And we can show that the best 3D model is from insect and mammal of the innate system. So to summarize this part, I showed you that tulips are spread on many phyla, non-venomous. I showed you that successful fold is used and reused in a broad context. I showed you evidence for conversion evolution, you know, the conus and the bee. I showed you also that machine learning are very useful in this aspect, and ClanTox is a discovery platform for novel and overlooked function. The last part, just if I have another few minutes, it's good. The last part is really to go even more courageous and discover new function. So where are we going to look for new function? Let me just show you and tell you that toxin-like sequences in mammals, I'm looking now in mammals, not in bee, not in insect, not in amphibia, and not even in, but in human. Can we find this using the tricks that I kind of shared with you? So about 10 or 15 years ago, there was a sporadic finding of endogenous, what they call prototoxin, named of lynx. This is a paper in Neuron from really early days, 2002. Novel modulator of neuronal nicotinic receptor associated with endogenous prototoxin lynx1. Those guys are expert in acetyl receptor, and they looked for something that will modify the receptor, and they found it. But now, with the tools that I told you, ClanTox, we can go to the deep sequencing of mouse and use data that were completely uncharacterized and look whether we can find tulips there. And this is the data from Phantom, 5,000 of them. We found 16 tulips. This is another one. We found 40 tulips in mouse. So who are they? What are they that we never heard about? So I'll tell you what's common to all those findings. They are rich in brain and in the immune system. All of them have signal peptides, means secreted. Many of them work in developmental signal. They come and go. Many of them are responsible to changes, and they are up and down in cancer and when the cells are disturbed. Many of them have a restricted expression. They are expressed at a certain time, certain organ, and not all over. And many of them have a multiple duplication in their genome. This is a common feature for all the tulips that I'm talking about in mammals. So let me just go very briefly on 3 case studies, and I'll do it very briefly. The first one is a protein called a Scrapie-responsive gene. This is a gene of 100 amino acids. If you take 25 out because of the signal peptide, it's about 60 or 50 or something like that, expressed in brain and induced in mice after they were inoculated with mad cow prion. This protein is extremely responsive. So, for example, an animal that were under social threat jump up. A case in which they were infected with some some strain of candida, they jump up. So they are responsive. The second thing that I want to show you is their expression. This is the gene that I'm talking about. It's expressed in a very interesting region This is, by the way, the expression of links. This is the hippocampus, the area that is responsible for memory and navigation and memory and so on in the mouse brain. So it's a very restricted expression. Probably its work also as a marker for autophagy. Let's go to case 2 that we worked much about. This is a set of what we called ANLP, alpha neurotoxin-like proteins. This is the sequence, just 3 of them out of many that we found. And they are very similar to snake neurotoxin of the 3 fingers fold, 1, 2, and 3, with a strong hook of a cysteine bridge here. So as you can see, there are a lot of cysteine spread all over. And the 3 fingers proteins are protein snake neurotoxin. And links 1, links 2, having the same fold. So now we are back to the, and when we take cells, this is stem cell, and we make them neuron in the lab, we see that on the undifferentiated cell, they are not expressed. But once they are differentiated and they become neuron-like, the expression of ANLP jump up. It's also present in the mouse brain, adult mouse brain. We make a bioassay for nicotine receptor, and we found that ANLP3, we found a bunch of them. When we tag them, we transfected macrophage. We do a nicotine assay. We can see that it's a receptor modulator of the receptor. We can show also that by having the cell ANLP3 in macrophage, we change the profile of the cytokine. This jump up. This is the control. This is after ANLP. This jump up from here to here. This jump down from here to here, and so on. So the cytokine, and actually what we are doing by doing this is we remove the nicotine blockage of inflammatory signal. So what we are doing actually is ANLP is a modulator of the nicotine receptor, and its expression defines the inflammatory response of macrophages. ANLP4 do exactly the opposite. So again, we find a set of genes, modulator, that play all the way on this reaction. The last point that I want to mention is that they are in the genome. This is a browser. This is 1.1 million nucleotide. All of them are listed here. Here is the ANLP that we found. Here, 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 and here. There is an expansion in the genome of those. Okay? So we have 9 or 10 of them expanded when you compare 2 genomes of rat and mouse. So what we show is that this is the links, and this is the toxin. This is activin. This is a big fold, and this is a super family. And we were able to add ANLP to this family, actually, in which in the mouse we have 10, and in the human we have 4. Just to wrap up, saying that ANLP belongs to a much larger structural superfamily, and you can see it here. This is the fold. And what do they do? We don't know much, but very interestingly, first of all, they appear as a bunch in the genome, tuck, 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 like duplicated. And in each and every cell of the immune system, there is a subset of them. So they are kind of a recognition password for those cells. So they are like an identity measure. So this is a very nice idea that the defense line of the immune system are marked by this very unique, and this is another proof for an expansion. So I showed you, I'm wrapping up, I show you the toxin, how we move from the toxin to other function and to this huge function. This is the amount of these guys, and a very large part in which many TOLIPs for the immune and the brain cell modulation. This is just to show you that they are very important in the clinic. This one is working on psoriasis, this on pain, this is on the immune system that I just mentioned, all those. This is on Alzheimer, this is links in the brain, and they are all sharing this structural identity. Is there more creativity in those guys? The answer is yes. Sometimes you have a very small deletion here, so alternative splice is working. Also, heterodimerization and cross-linking between two of those are also found. So a lot of creativity on these guys. So I'll skip this just to summarize this TOLIP story. From the methodology, I showed you that the machine learning predictor was very useful. From the discovery, I showed you that we can find non-venom TOLIPs in diverse phyla, including human. From the biological impact, I showed you a mimicry of animal toxin for extended cellular function. I didn't show you much about antibacterial channel modulator. I showed you quite a bit. And the validation is tested TOLIPs act as a bioactive endogenous modulator. So what have we learned from our scorpion, snake, and hydra? This is the real toxin, and this is the TOLIPs. The real toxin, they have receptor as target, yes. They are very short. In case of human and mouse, they are a little bit longer, so they are not that short. Simple modification, those are rich in modification. Do they have a chromosomal amplification? Yes. But those guys often use irreversible action. The one that I told you are reversible. And this is the major difference between TOLIP and toxins. So closing remark. I showed you in the beginning of the talk why maps are important to follow the evolution footstep. I showed you how innovation on one hand and replicators, just copy what you have, is a good idea. Plasticity in brain and in immune system calls for innovation. And there is a unification function, which is a common phenomenon. And just to say, there are a lot of many good people that helps. This is just some people that left the lab, and this is from this morning when I tried to dig my car here. But that's good. That's a new experience. And of course, you can have more reading as much as you want. And thank you so much for your attention.
I have two questions. The first question is, when you use the map for function prediction, so you use evaluate, but actually most of the sequence clustering programs, they use sequence identity.
Mm-hmm.
Do you find there's a specific advantage of using evaluate instead of sequence identity for function prediction?
Yeah.
And,
yeah.
And the second question is just how you do the clustering.
What's the second question?
Just how you do the clustering. So
how do you do the clustering? Okay. Mm-hmm. So the first question was concerning the e-value that are used. So I must say that intuitionally, by intuition, we wanted to use very modest e-value initially, like 1 or 10 to the minus 2 or whatever, something. But then we just played. I mean, one student say, let's do 20, let's do 100, let's do 1,000. And we figure out that no matter what, if you have enough data, those small, very noisy, I mean, it's noisy like hell, because by doing e-value of 1,000, it means that you have, on average, by random, 1,000 wrong hits. Not good hits, wrong hits. Never mind. If you have millions, it can manage. And the trick is really to do the merging very slowly. And the merging is the trick. You do bottom up. So you build up a very, let's say, solid type of information. And as I showed you in one of the slides, in each and every step of merging, you are comparing all the options of merging, and you pick the best. So some of the merging could be valid, but they will wait. So you do the merging, the most accurate one immediately, and immediately you do the re-averaging. And the average capture all those consistent. So it's a very elegant and very simple, by the way, idea. Now, the merging itself that you asked for the question, for the second question, I don't know if I think I have so the merging, for example, we didn't know, right? How should we know how to merge two clusters? So we tried many things. we try to do, average of arithmetic and arithmetic means and geometric and harmonic and Hausdorff and then all kind of, and we have a way to check, as I told you. So we take Go annotation, PFAM, scope, class, and we ask which one is giving better or worse. And we figure out by doing all this analysis that some merging are better than others, and we stick to this. So once we had confidence in our merging, we put it aside. We don't care anymore about PFAM or whatever. We just do it because it gives us a better solution. So this is just to show you that we really took very seriously this aspect of yeah. So I hope
I guess I didn't answer your question very clearly. The first question is, so you use e-value,
yes.
but most other sequence clustering programs use sequence identity. So is there a specific advantage of using e-value instead of sequence identity for this kind of
e-value instead of sequence identity, you mean?
Yes.
Yeah. So, okay, so again, in the very beginning days, which really goes 20 years ago, we did all the tests. For example, we did BLAST, but we did also Smith-Waterman. We did also the direct bit, like sequence identity as a measure, and figure out that the e-value is far better than
what do you mean by
percentage,
better?
any percentage or any, because, but this is legitimate if and only if you are using huge data. If you use small set, don't even dare to use e-value. So e-value was legitimate only because we moved from thousands to millions. And there is, I suggest you read, there is one paper in which we discuss it in fairly details in 2008 or so.
So in your machine learning algorithm, there were many features that were sort of predicting structural aspects about the protein from sequence information. Do you think that there's any promise in incorporating that into your clustering approach as well, or do you think it'll just add noise to the prediction?
Okay, so you're asking a very good question about the feature being also structural based. So basically, you are right. I mean, when I said it's very important to do feature engineering, I mean, think first what is really that you try to capture. And in our case, we really try to capture structure. We try to capture something that is very strange. I mean, we don't care which fold, but we know that it has to be almost circular, almost a ball, right? How you put feature to such sequence that you want it to be a ball, right? So that was the idea, and I showed you a few examples of how we do it. But you're right. The first protonet, the map that I told you, was used very successfully for structural people. Why? We were involved in really many years ago in what they call target selection. What is the best target that one can pick from an unknown fold so that you have a higher chance of finding new fold that were never checked before? So the only things that we can provide was to go over the tree and assign path or scope on top of the tree. And then we saw that some niche in the tree were completely empty. Empty, like dark. And we said, why is it? So I'll tell you two reasons why. First, we didn't know. We just looked at it. And the map became, some was, with light, and some remained dark, because we couldn't find anything there. So the idea was, for the target selection people, go to the dark area, pick one, and you'll have a new fold. However, we figure out that there was another explanation. There were two. One, which is what I just said, The other, they were extremely rich in membrane proteins that were very underrepresented in PDB. So we were able to basically look at holes in this, type of islands in which we have either very difficult proteins for crystallization or for any other, and in addition, area that was neglected. And what we did, just because you asked, and it's an excellent question, we made another kind of measure. How far you have to travel in the ways, travel, in order to get from one structure to another. If it's a long distance, you know that you have something in between. So your intuition is good about the structure. It's very informative. Please.
Could you say something more about the biological function of these things potentially in the mammals?
Yeah.
Scorpions, I think I could get.
No. So actually, I think what we have shown, and that we have basically 3 or 4 major functions, just to summarize it without the details. One is proteins that are extremely small, that works as modulators, so really allosteric elements. They just plugged in and compete with the regular nicotine, or the regular acetylcholine. This is what we showed. That indeed, they just stay there, and by that, the nicotine pocket is half blocked or do something else. So they are really very elegant way to affect major channel and receptor. That's one. Second, in the immune system. In the immune system, they are working very nicely as recognition because they are like tags. I have it, I don't have it. I have it, I don't have it. So they are like markers, small tag in your book note. It's easy to produce, no big deal. Some of them have a GPI, so they are stuck in the membrane. So they are marker, and they are beautiful on this level. The other, so this is the second. The other extremely important one is the antibacterial. So a lot of the protein like WAP that I jumped over are really antibacterial function. So they can either make holes in the bacteria, in the membrane, or they do it in some magic way, but they really affect some specific bacteria, such as defensive, and those are peptides that are floating our system, our epithelial system. So if you look at all those and what's common in between, you can see that those are proteins that needs to be kind of modulator or protecting agent, and they need to be in a high concentration. And they need to be easy to produce and stable. So who is in the world that's so good in it? Scorpion. Yeah. This is the other feature that I mentioned, that they are very responsive. So whenever the cells is flipping itself, boom, they are on or off. So this is, I really like the design. As I mentioned, as I showed you, even maybe I'll show you just one more slide that I jumped over, this one. This is an area of 0.5 million, so 500,000. And everything that is marked here is a version of this specific fold. So you produce many in the genome. Each of them is a little bit different. So you have a battery now. A battery of what? Anti-inflammatory activity. You see? So this is another function. So I think there is a commonality. And this will be the same, by the way, if you go to weird genome. And we have few, including bacteria, including nematostella that is, like from the sea. You see this repeating. So I think it's the principle. Yeah, exactly. Yeah.
Well, thanks so much. It's fascinating.
Thank you very much. Fantastic. I really enjoyed.